Hi, everyone. I'm Nicole Carter, as you guys know. I've been working in Dr. Tom Gilmore's lab in the biology department for the past 3 years. And today I'm going to talk to you about our work exploring the role of transcription factor NF-kB in coral bleaching. So first of all, what is coral bleaching? So corals are a member of a group of animals called phylum Cnidaria, which also includes sea anemones, jellyfish, and hydra. And like many other members of this phylum, corals undergo what's called symbiosis with algae. So by symbiosis, we mean that this is a mutually beneficial relationship wherein the algae literally live inside of the host coral. And that's what gives this coral that you see on the left, this nice brown healthy color here, that comes from the algae. And the algae are really important because they provide a vital source of nutrients for the host coral. And this relationship is also beneficial to the algae in that the coral provides them with a place to live. So that's really important. However, in what we call bleaching, we actually see the loss of this symbiosis. So the algae are expelled from the host coral, and that's what you see on the right here. You'll notice this coral is kind of that white color, and that's because those algae have been expelled, and we call that bleaching. And if this is a really widespread problem, it's currently estimated that about 90% of all corals have experienced some bleaching. And so while we know that this is associated with things like rising ocean temperatures and ocean acidification, we don't know a whole lot about what's occurring on the molecular level. And that's what our work is really trying to figure out. But first of all, why is this important? So coral reefs are really important sources of biodiversity. It's estimated that corals support 25% of all marine life and more than 2 million different species. And this is despite the fact that they cover only 1% of the Earth's marine environment. Coral reefs also have huge economic value. So for example, tourism to Australia's Great Barrier Reef generates more than $5.7 billion per year and provides full-time employment to 69,000 people. And corals are also really valuable sources of pharmaceutical compounds. So this is actually the structure of a drug called Halovin, which is used to treat breast cancer, and that was derived from a coral reef residing sea sponge. So another term that I threw out in my introduction is a transcription factor. So what does that mean? So what I'm showing you here is what we call the central dogma of biology. And basically what that means is that in every cell in your body, you have DNA. And all the cells in your body have the same DNA. But as we know, a hair cell looks a lot different than a skin cell, and that's a lot different from a blood cell. And that's because at any given time, most of the genes are not actually on. They're in what we call the off state. But then in this step called transcription, the DNA is used as a template to make an intermediate called RNA. And then in another step called translation, that RNA is used as a template for protein. And this is a highly regulated process. And one of the ways that this is regulated are through proteins that we call transcription factors. So transcription factors are going to bind to the DNA upstream of a specific gene, and they're going to either activate or repress transcription of that gene. So they're going to cause this step to happen. So my lab focuses on a specific transcription factor called NF-kB, which is involved in a lot of human diseases, including cancer and autoimmune diseases. Alternatively, if you had too little NF-kB or your NF-kB was not working, you would probably have immunodeficiency. So NF-kB can be regulated by a number of upstream signals, which might include bacteria or viruses, cellular stress or antigens, which are just proteins presented by foreign molecules. And NF-kB can activate genes that are involved in things like innate immunity and inflammation, and then adaptive immunity, so basically an immune response. So how this relates to coral bleaching is that primitive organisms like corals actually have NF-kB, and the pathway is really highly evolutionarily conserved between these primitive organisms and humans. Furthermore, because NF-kB is a regulator of immune response, and previous research has shown that coral bleaching is associated with upregulation of immune activity, we think that it would logically make sense if NF-kB is involved in bleaching. So another thing I need to tell you is that while I've talked a lot about corals so far, I'm not actually working directly with corals in the lab. I'm working with a sea anemone called Aiptasia pallida, which you see here. And so there's a few reasons that we're doing this. So corals are actually really difficult to work with in a laboratory environment, and one reason for this is they have a really hard exterior that makes it difficult to do simple experiments like a tissue extraction. Alternatively, my sea anemones are soft. I can just grind them up in a test tube and get pretty much whatever I need out of them. I don't know why that's that funny. I'm glad, okay, I've kept everyone awake. So they're really useful in that regard. Another thing that's nice about them is that corals grow really slowly in the lab, and once we induce loss of symbiosis, so kick those symbionts out, they usually die pretty quickly. Whereas our sea anemones have tentacles, which allow us to just feed them, and they can stay alive for a while in what we call the aposymbiotic or bleached state. So it's without those symbionts. They're an appropriate model organism because they're evolutionarily closely related to corals, and they harbor the same algal symbionts. So here you have a symbiotic anemone. Again, you'll notice has that nice brown, healthy color. And then here you have a bleached anemone. And these little brown specks you see are actually algae that has been expelled from the anemone. Okay, so we're studying the role of NF-kB in loss of symbiosis, or bleaching, using these anemones. So one of the first experiments that I did was I wanted to see how are NF-kB RNA levels changing during bleaching. So again, here's the central dogma, just for a reminder, and the experiment that I did here is called a qPCR, which measures levels of RNA, which is this intermediate that you see here. So presumably, if you have higher levels of RNA, you're going to have higher levels of protein. And if we have higher levels of protein, we have higher levels of NF-kB activity. So what I did was I start with symbiotic and aposymbiotic or bleached anemones. I extract RNA out of them just by, grinding up in a test tube. And I do this qPCR experiment. And so this is a time course experiment that we did where we bleached anemones for different amounts of time, and we measured NF-kB levels. And what we see here is that NF-kB levels increase during bleaching and peak at around 3 days, by which point NF-kB RNA levels are 3.3 times higher than the levels of NF-kB in the symbiotic state. And we were also able to use this technique to measure loss of symbiosis. So here I did the same type of experiment, but I did qPCR for a different gene, which comes from the symbiote. And we'll see that at day 1, we have about 80% loss of symbiosis. And then by day 3, we're down to 99.6% loss of symbiosis. So what we see here is that NF-kB mRNA levels increase during bleaching, and this correlates with loss of symbiosis. So other data in our lab, which I personally was not responsible for collecting, shows that protein levels also increase during this time period. So we've shown you so far that there is a correlation between NF-kB, RNA, and protein levels and loss of symbiosis or bleaching. So then we want to answer the question of, is NF-kB activating a specific gene called the Inducible Nitric Oxide Synthase gene, or iNOS? And one reason that we thought that NF-kB might be activating this is because this is a target gene of NF-kB in humans. And other research has shown that this gene suggested that this gene might be involved in bleaching. So what I'm showing you here is a diagram of a specific gene, or of a typical gene. So this region right here is called the promoter region. That's where your transcription factors like NF-kB are actually going to bind. And then this right here is what we call the coding region. So this is what actually is used as a template for RNA. And the experiment that I did here is called a luciferase assay. So what I did was I identified that promoter region for the iNOS gene. I made an artificial DNA construct where I put that gene next to it, or that promoter next to a gene called luciferase, which produces a light signal. And then I performed these experiments in human cells. So by measuring that light signal, we can tell whether or not our transcription factor is activating the gene. So it's just going to bind, produce a light signal that we can measure on a machine. So interestingly, we found that NF-kB does not actually activate the iNOS promoter in Aiptasia in our assays. So here's what I really want you to focus on. So this is the Aiptasia NOS promoter, that diagram that I showed you on the previous slide. And what you'll see here is that regardless of whether or not we add NF-kB, the level of light signal that we see, which we call luminescence, is about the same. Alternatively, when we use a synthetic promoter that our lab designed specifically for NF-kB to bind to, we do see higher levels of luminescence. So this tells us that our anemone's NF-kB is not activating this promoter. And then we followed this up by doing another qPCR experiment, and we showed that levels of this gene are actually not increased in aposymbiotic anemones. So our conclusions, today I've shown you that NF-kB, RNA, and protein levels increase during bleaching. I've also optimized a quantitative approach to measuring symbiote levels during bleaching experiments using qPCR. And we found that NF-kB does not activate that iNOS promoter during bleaching. And then future directions, while I've shown you a strong association or correlation, as we all know, correlation does not mean causation. So we want to know, is NF-kB causing bleaching? Is it a response to bleaching? Or is it neither? And then we also want to know, since NF-kB does not appear to be activating that iNOS gene, what actually is NF-kB activating in Aiptasia? So with that, I'd like to thank my lab, our funders, especially the Arnold and Mabel Beckman Foundation, who have been paying me the past 2 years, and of course, KHC and all the lovely faculty and students in the program. So with that, I'll take any questions. Thank you. Any questions? They could be sciencey or not sciencey. Jackie!
Regarding your QPSR data, can you explain some of the, the significance, like the p-values for some of them?
Yeah, so I don't have the p-values in the captions for this slide, but if we go to, I guess I didn't, okay, yeah. So here, all of these are statistically significant compared to the symbiotic anemones at the level of less than 0.05. I believe this one was less than 0.01, but I don't know it offhand. In my original figure for my actual thesis, there's asterisks in there that show that. And then these are all like really statistically significant, like less than 0.001. And then here, this activation was significant, but this little tiny bit of activation was not. Yeah?
This is just like an out of the blue question.
Go for it.
Because in marine bio, we learned that the Affinity Lab studies like corals on mangroves, lilies. Like, does this project have anything to do with that, or is it just that the Affinity Lab provides your sample?
So, yeah, so they're actually our collaborators. So they have collaborated with us in a lot of the anemone stuff, helping us to like get the anemones set up in the lab. They did some of the more bioinformatics experiments relating to this project, which isn't data that I showed, but yeah, they're our collaborators. Although we're not working directly with corals, that's sort of a project that I guess they're getting started, but we're working mainly with anemones. Yeah?
So if hypothetically somewhere down the line you did find highly suggestive evidence that like NF-kB works, like would you be able to do some sort of gene recombination in order to like, I don't know, stop coral bleaching?
You mean as far as gene editing in the lab or in?
Like, no, in the environment.
Yeah, potentially. So yeah, again, that would probably be pretty down the line, but there are NF-kB inhibitors that are commercially available. So theoretically, if we were able to find an inhibitor that does work for our anemones NF-kB and that also works for coral NF-kB, if we could find some way to treat corals with that, that might be able to halt bleaching. Again, the actual delivery of that would probably be a little bit difficult. That would take some engineering, but yeah, that's definitely something that we're considering.
Would you do it by like releasing inhibitors, like releasing the, I don't know, how would it even work? Like how would you insert that into the environment?
That's something, yes, that's something we still have to work out. Another possibility is, so there's talk of what's called like designer corals. So there's people that are studying what makes some corals more resilient to bleaching than others under the same conditions. Like some corals might bleach and others won't. And people are looking at genetically what is different between those. So again, this is probably way down the line, but if we could genetically engineer corals in the lab that are more resilient to bleaching, maybe if we did something, I don't know, with their NF-kB promoter that makes it less active, and like we put those back into nature, maybe we could somehow do coral rehabilitation that way.
That's all we have time for today.